Superbugs are a worldwide health problem that antibiotics can't control. A recent study found that bacteria could genetically transfer their antibiotic resistance to other bacterial species, thus turning less dangerous bacteria into antibiotic-resistant superbugs. The research study focused on two patients, patient 1 and patient 2. Patient 1 entered a hospital with an intestinal infection caused by Enterobacter cloacae, or E. cloacae, a bacterial species known to be resistant to multiple antibiotics. The reason for antibiotic resistance is found inside bacterial genetic structures called plasmids. Plasmids carry the code used to produce KPC, an enzyme that neutralizes antibiotics. As expected, patient 1's infection did not respond to standard antibiotics, and more powerful drugs were needed. Patient 2 entered the same hospital with a respiratory infection caused by the bacteria Serratia marcescens, or S. marcescens. Initially, patient 2's respiratory infection responded to antibiotics, but then ceased to respond to treatment. Patient 2 became gravely ill. Here is what is believed to have happened. Tests determined that E. cloacae had somehow spread from patient 1 to patient 2. Patient 2 is now infected with two bacteria, antibiotic-resistant E. cloacae in the intestines and antibiotic-resistant S. marcescens in the lungs. E. cloacae entered patient 2 and settled in the intestines in close proximity to S. marcescens. Unexpectedly, the two different bacterial species could and did interact. E. cloacae attached to S. marcescens and used a tube-like structure, or pilus, to transfer plasmids with the antibiotic-resistant trait to S. marcescens. S. marcescens incorporated the antibiotic-resistant trait into its genetic code before reproducing. Antibiotic-resistant S. marcescens spread throughout patient 2's system, then colonized in patient 2's lungs that had been damaged by the previous infection. A case study review determined that healthcare professionals or visitors could have been involved in the transfer of E. cloacae from patient 1 to patient 2. This type of research has led to the development of protocols for healthcare facilities. These protocols should decrease the incidence of antibiotic resistance that leads to the creation of superbugs. The protocols include isolating patients with known antibiotic-resistant diseases, mandating that equipment used to treat a patient stays in that room, and most importantly, requiring healthcare professionals and encouraging visitors to foam or wash their hands upon entering a patient's room and upon leaving the room. This simple practice is a huge step in preventing the spread of bacterial disease and the creation of superbugs in healthcare facilities. For more information on this subject, go to the Center for Disease Control website address shown here.